to start my story, I should probably talk a bit more about where I've come from and why I'm here in Berkeley today. So, I actually am, well, I've been in Toronto for the last 9 years, but I'm born and raised in Bangladesh, born in a very small town that most people from Bangladesh probably won't be able to name either. And yeah, but I was always like from, I guess, when I was a kid, I was really always interested in science, loved school. So was a geek that didn't, I mean, wasn't very cool in school. Back then, being nerdy wasn't cool, but now I think it's not bad at all. So I loved science. I really want, but I was never sure about what I wanted to do with it. But I did realize that being in a small part of Southeast Asia wasn't going to expose me to the kind of innovation and opportunities that I thought I just wanted to be involved in. So for I guess at the age of 18, I moved to Canada for my undergrad. And yeah, I was the first person in my extended family to move at an early age for undergrad. And it was a really cool ride. I When I moved to Toronto, I tried to, like, obviously started with a biological chemistry specialist program and started to get my hands dirty in a lot of research labs. Tried a lot of things that I didn't enjoy, but somehow in my second year, my PI, who is actually the co-founder of my our company, it was or he took second year organic chemistry, and he was just the most passionate and excited man about chemistry and how he's going to change the world with chemistry. So I think his passion was it was it was just so exciting to see someone so excited about science. And I eventually figured out how to do research with him. So for my undergrad, I did a couple of research projects, and from then on, I guess there was no stopping. Just started a direct entry PhD to keep continuing the work. But I guess in what I did see was even as just an undergrad student doing research at a big research lab, I got to see his startup company spin out right next to me. So the fume hood where I'm working and trying to make my small molecules, doing my purification overnight, and I see the next fume hood is his first startup, which it started with one employee right next to me, but eventually became 100 employees in three different locations. So I think from my early 20s, I got to at least see entrepreneurship and science entrepreneurship. And my father has always been an entrepreneur, but in the fashion and manufacturing industry. So I could never correlate, but I think at this age, I see that there's a few things to learn from him as well. But it was really cool seeing entrepreneurship just right next to me through on my undergrad and my PhD. And yeah, started off looking at HDAX 6 inhibitors, and because there's a lot of HDAC targeting. It's just a class of proteins that are involved in a couple of diseases, most notably known in cancers. And there were a few drugs in the market that were targeting these proteins already. There's actually 4 FDA approved ones, but they're very toxic, can only be used for relapsed cancer patients, can never be repurposed for other diseases. So there was a huge need to make better drugs. And that's the project I started to do a lot of chemistry, and then the molecules I designed would do love some cellular biology work. Got to, because I was in decently big, I think at least 30 people research lab. There was a lot of collaborative work as well. So I think I started to collaborate with at least PIs from 7, 8 countries. So all over the globe, we had a lot, and that led to quite a few publications. But that also allowed me to learn a bit of networking in the science space and look at other people's work, figure out how to collaborate, how to entice another professor to come and work with us. And yeah, I think once after a couple of years of a lot of science and a lot of failures, and I'm sure that's something that's the same across this whole table. And most of the people here have done PhDs, so you know that it's mostly 10 failed experiments and then maybe one that has worked. So that was my journey as well. And eventually, after a few years, we started to file some patents, and we really felt like there's some differentiation compared to what's out there and what is being investigated in humans right now in clinical trials. So at the tail end of my PhD, well, my PI wanted to spin out the company a long time ago, but really at the tail end of my PhD, when I also thought like, okay, like I think we can truly make a difference now and maybe expand up outside academic grants and try to scale this into something that could really help patients. That's when we spun out the company, exclusively licensed out our IP from the university, and raised pre-seed mostly in Canada, a bit in the West Coast as well. And yeah, so I'm a first-time founder. Learned a lot of things on the go. My PI, yeah, this is the fifth company out of our academic lab. So I do have a little bit of help from a serial entrepreneur, but my PI is I mean, he's not usually as involved. So there was a lot of things to learn on the go. And I mean, he wasn't very involved even within the academic lab. So we were kind of left alone. But that, I think, made us very independent to figure out what project to work on when we were in our PhD, and now um, how to figure out the business and what to do next and how to reach next inflection points. So a little bit about HDAX Therapeutics now. We design targeted therapeutics for neurological diseases that have axon transport dysfunction. So specifically starting with the first indication of chemotherapy-induced neuropathy in cancer patients. And we are trying, when these patients actually, there's no real treatment for these patients. They only depend on symptom management. So you're looking at opioids, painkillers, which don't really target the nerve damage that's actually happening in these patients. So we are trying to develop the first ever disease modifying therapy for neuropathies, starting with cancer patients who have neuropathy from the toxic chemo. And honestly, all the toxic cancer drugs, even the newer ones they're getting. And eventually we want to expand towards other neuropathies and other also other diseases are their secondary indications of interest that are impacted by the protein HDAX6 that we're targeting, hence the name HDAX Therapeutics. And I think 3 months ago, we became part of Berkeley Skydeck. So we are a Berkeley Skydeck fund invested company, which is why I spend a lot of time in Berkeley now. Probably here for the next 2 months. So happy to meet if you're in the neighborhood. I'm usually here.
Hey, Nabanita, I'm going to jump in and ask some questions. You mentioned a couple of things. One is you were able to entice a bunch of professors to work with you. Tell us how you were able to bring professors on board to work with you. These are, I'm guessing, mostly academic focused, or maybe you can give some more context.
Yeah, well, that's actually a tough question. I think it was more like a chain reaction. Like, we have some collaborators. So we're a very medicinal chemistry. I'm a chemist by training. So we don't do a lot of the biological or the deeper biological work, the in vivo work. So we usually try to either outsource it from CROs, which is extremely expensive, or if you can get a collaborator who's going to work with you, we figure out what compound's going to work well, and then you test it in vivo. That's kind of like our sweet spot. So I think you just reach out to a lot of people, talk to them about the exciting science you're doing, and I think that's where I gained a lot of confidence in reaching out to high profile PIs and talking to them about the science, getting them excited so that they would want to collaborate with us. And a lot of the times it didn't work out, but some of those times that it worked out, it ended up being pretty great. And then afterwards, it just becomes they know more PIs, right? So eventually you start, and I mean, someone else wants to do one set of experiments, and then suddenly you have 7 PIs in one publication, which is not the best, but also you also get to have a huge network of really high profile investigators who do really good science.
Yeah, congratulations on that. And how did you ever think about the risk? The risk in this space is pretty high, of failure is high. What was your decision-making process in that, in starting a company? Because you said your PI wanted to launch a company a long time ago, but then what were you thinking? And what was that like? point where you said like, I need to start a company?
So I don't think I I I woke up and I was like, oh, I think I'm a born entrepreneur. This is exactly what I want to do in life. I just wanted to do really meaningful work and just be at the forefront of innovation and have a more of a leadership role and have a say in how it's working. But with my PI, like, and I think we took a considerable amount of time. Like, I think we built the science with many millions in grants, many years before we decided, okay, now we can spin it out. But because my PI was entrepreneurial and he had pharma and VC connections, he would just talk about our science here and there, and he would kind of like, oh, let's spin it out. J&J is talking about it. And we're like, no, we're not ready. So only when we really felt like we're ready, and I'm also looking for what to do next. I think when the science really reached its inflection point is when we thought when it could no longer be pushed by just academic grants, and it just needed uh, more people involved and definitely more capital involved. That's when we decided to push it out. And I don't know, in that the whole just building the business process, I got to know so many people. I don't think I thought very much about what I was doing, which is probably a good thing. I didn't think about all the risks. But if I thought too much, I probably wouldn't have done it because it is extremely risky and very stressful.
Good, cool. Thank you so much.
Can I ask something follow up? How do you think about like the risk and not getting scooped? And you know, you were saying how your professor would just talk about it, so he didn't worry about confidentiality as much.
I mean, in reality, the most confidential thing is really like the microstructure, the chemical structures, what we work with. And I mean, as long as you don't disclose any of that, and I mean, it's very similar to how you're working in your academic lab and building the science. And before you publish, you probably don't want anyone knowing about the nitty-gritties of your project. This is similar, but with a lot more risk, I think. It's the same analogy, but we have multiple legal teams and we're more cautious. And sometimes if I mess up a little, my co-founder, someone in the meeting, Zoom meeting, is gonna DM me like, Nap, shut up. So yeah, so definitely making some mistakes here and there, but nothing that could at least like jeopardize the company, I think.
Awesome. Thank you. If anyone else has questions, you can jump in now, or we can take them towards the end. Okay. Next, we're going to hear from Adam. Adam Schieferecke, I hope I said it right. So he is a co-founder at a stealth mode startup, and you can probably tell us a little bit about what that is. He is actually a candidate in the lab of Professor David Schaefer at UC Berkeley, where he pursues basic and translational gene and cell therapy research with the goal of expanding human knowledge and improving patient lives. Accordingly, Adam recently co-founded a stealth mode biotech startup that applies his prior academic work to develop therapeutics for cancers of unmet need. And I met him because he started this biotech at Berkeley organization. I think this was mainly focused for the graduate population and especially those who are interested in starting up. So we're excited to hear from Adam, his journey, and yeah, everything he can share with us. Thanks.
Yeah, thanks so much for the introduction. So yeah, currently I'm in my last year of a PhD program in molecular and cell biology here at Berkeley, and also in my first year of spinning the work out of the lab in pursuit of making cancer therapies for patients. And it all kind of started out, I grew up in rural Kansas and went to the state land grant institution there for a BSMS program. And that's where I realized my passion for science when studying evolution and virology, doing research. And Kansas State's really unique in the sense that it operates some high containment labs and is adjacent to a BSL-4 facility. So this exposed me to some pretty talented virologists. But a summer fellowship in the virology and gene therapy track at Mayo Clinic kind of helped me realize that I was more interested in the good viruses. So like these non-pathogenic viruses that scientists and doctors have harnessed as therapeutic gene delivery vectors that can support healthy cells or selectively eliminate cancer cells. And so I came out to do my PhD in Dave Schaefer's lab here at Berkeley. And some of you might know Dave has a pretty impressive reputation and track record with spinning out successful companies from the university. And he, stuff that was developed in his lab is currently in 7 active clinical trials. And so despite all of this, I kind of came into the PhD program with a pretty open mind. I wasn't, set on being an entrepreneur, and really kind of explored things. And I would say that my process of getting here was through a process of elimination. So for example, like teaching is just one of the most fundamentally important jobs in society, but when I taught a pre-med weed-out course at Berkeley, that led me to decide I'd prefer maybe not to have teaching as my full-time job. And the pandemic being in the middle of my PhD led me to realize that I really like working with people. I really like doing the scientific research with colleagues. So I really wanted to be physically in the lab, not remote. And, some experience I have working with public policy kind of led me to realize that that work is also very important, but it stresses me out. So I'm, happiest doing the research. So by kind of early in the 4th year, I had narrowed it down to being an academic professor or spinning out a company or being this nomadic tree person who sustains himself by gathering nuts and berries in the woods. And I'm guessing that there's probably more folks watching this who are considering the first 2 options. So I'll focus kind of more on comparisons with those. But one of the things that I really value and that I was scared to lose when going out of academia was kind of that sense of intellectual autonomy to pursue new ideas. And in other words, like above all else, I didn't want to be just like a cog in the machine. And I was really surprised to find that there's actually a lot of parallels between when you start a company and when you're like starting an academic lab, especially on the scientific side, in the sense that, you're applying for funding, you're building a team, you're driving the science. I mean, the science has to be through more of an applied lens rather than a basic lens, but you're still, having that kind of autonomy. And so I've really had a lot of fun so far with doing this, and I'm really certain that this is kind of the right path for me. And so, yeah, so essentially this was based on, I was working on a couple projects in the lab, and one of them just kind of had more clear commercializable potential than the other, and it started working really well. And so we decided to start the company, and with the co-founder, I had been working on an academic project with a postdoc in the group, and we knew that we kind of worked together and we had complementary skill sets, and so he went in on it, and yeah, and it's just been really fun so far. And so I think, so why spin a company out of academia? I think the basic research is fundamentally important for creating new ideas and the underlying knowledge needed to drive innovation. And without basic research, we would have absolutely nothing. So we have to fund basic research robustly and have people that do it. But at the same time, the basic research alone does not create value for society without the people who are willing to translate it into products or therapies or whatever it is. And so, you know, for better or worse, in this country, the primary way, and really the only way for some cases, to translate this basic knowledge into products that bring value to society is through a company. And so for us, that's really focused on, you know, using some of these discoveries and this platform technology that we've developed in the lab to producing therapies that give patients more life. And so, you know, in the therapeutic space, this means that you have to go through this whole process that you just wouldn't do in an academic lab, which is like carefully selecting your approach, taking into consideration things like GMP manufacturing and, you know, what is the FDA is going to like, what's been clinically validated before. These are things you wouldn't think about if you're just doing it academically all the time. And then actually generating this IND enabling data that get you approved to start a phase 1 clinical trial, and then performing through your clinical trials, which take an increasing amount of capital as you go through them to ultimately, hopefully get FDA approval for the products that you're working on. And so I guess the last thing that I'll just talk about before I take questions is just some of the resources that I found really helpful while I was here. I will say, you know, like Naresh has a really great thing going with this BPEP, and this has been really awesome. You know, he kind of started this when there wasn't really anything too much around in the ecosystem. And so this is definitely a great resource. And I know that you're all already familiar with that since you're here. But in the last 5 years, I'd say that it's really, you know, the system or the ecosystem has really grown and it's really developed. And I think there's going to be a lot more development going forward in the next 5 years as well. And some of these big developments are like the Baker Bioingenuity Hub and Baker Labs. So this is spin out incubator space that you can get to, you know, as kind of a next stage when you're ready to get your own lab outside of the university, especially if you're in the bio space. You can get that before you're ready to go, you know, get a bigger place or whatnot. So this is a really great resource, and they have a lot of events there, and they're even you know, trying to get set up some internships for graduate students, and this will be really great. And then there's also the Life Sciences Entrepreneurship Center, which is a great place to kind of start. That's lsec.berkeley.edu. And Darren Cook has been a great resource there where he can kind of help you connect with resources or get you started if you just don't know where to start. And then biotech at Berkeley as well, if you're a graduate student interested in the biotech space, you know, this has been another great resource. And so, yeah, just as someone in the therapeutic space, I'll just pull that, put in those plugs for some of these resources that might be relevant for people who are also interested in it.
Awesome. Thank you so much, Adam, and for these resources as well. Do you want to tell us a little bit about why you'll decided to go stealth mode?
Oh yeah, I would say, you know, it's not like this is kind of like going back to the question about like what you share with people and whatnot, and I don't think it's such a big deal. I mean, I basically said what we do. We're making cancer therapeutics. That's kind of our focus. We're just in a very, very early stage, you know, and figuring some things out. So I think it gives you a little time to do that exploration before you're ready to go advertising yourself. So.
Cool. Thank you for those plugins, Adam. Like you mentioned a couple of good points. One is like you're having fun doing this. It's very, very important for people to understand that. I'm sure Nabanita can attest to that, and so Anders. And the second part is translation. We at deeptech, we try to convey this message to everyone that, if you're doing research, make sure you think about ways to translate that research to something more useful. Otherwise, it's going to end up in a pile of papers or a patent that nobody, including you, will care. So I think we can do better with science than that. Maybe you can talk about what was the toughest decision you had to make in terms of going into a startup as opposed to other careers? And who helped you? What was the thought process? Did you talk to people? What was that process looking like for you?
Yeah, well, I mean, there were just a lot of concerns, and I think some of them might be misconceptions. Some of them are also valid. I think some considerations I said, I mentioned like the intellectual autonomy part, and I've really found that to really be there with the startup, and being able to lead the science and do a team and build a team that's really been there. I think the startup is a little less, it's not like a 9 to 5. I mean, it can be, but there's some times where you just have to crunch to do things, and that's not necessarily for everybody. So that's something I'm kind of willing to do, but there's that, and then there's just the it's not like a set growth, like you set some milestones and then you don't grow until you meet those milestones. And so I think that's also part of what makes it really fun, I think. And yeah. Good points. And
I think I want to go back to one question Radhika asked, and Nabanita was mentioning about, like, if somebody's going to scoop your work, one thing we observed over the past 10 to 12 years at Berkeley is, like, if somebody can just copy your work but listen to you and go do something, there's no company for you guys, for any of the listeners here. It comes down to execution. It's a long road, and people need to remember that. So don't really worry too much about those, I think.
I will totally agree with that. And if your idea is, like, if you just say what you're doing and your idea is so simple that someone else could just hear what you're saying and go and recreate it, then it's probably not the best idea, right? So there's a lot that goes into it, and especially if you've been studying this and working on it so long, there's few people who could do it better than you. So I do think that, like, there's a lot of value in talking openly with it about people. And I think that keeping the mindset about, treat it as you would any other academic project that you haven't published on yet, and just talk about it at that level. I think that's pretty good advice.
Thank you, Adam and Naresh. We're going to keep a few more questions to the end, and then we're going to first hear from Professor Naar. So Professor Anders Naar is a professor of metabolic biology in the Department of Nutritional Sciences and Toxicology at UC Berkeley. He received a BS degree in biochemistry and biotechnology from the University of Lund in Sweden, and a PhD in molecular pathology at UC San Diego in 1995, where he studied nuclear hormone receptor mechanisms of gene regulation. He was a postdoc with Robert Titian at Berkeley, where he discovered the human-mediated transcriptional coactivator complex. The primary focus of Professor Naar's lab is to elucidate transcriptional and microRNA regulatory mechanisms that govern cholesterol and lipid metabolic homeostasis. So today we're going to hear from him about his experiences in academia and also as a founder of a therapeutic company. So without further ado, I'm going to pass it on to Professor Naar.
All right, thank you so much, Radhika, and thanks for having me. I'm, as Radhika said, or maybe you didn't say, I'm originally from Sweden. I actually grew up also, like Nabanita, in a small town in a forest, and there was pretty much nothing else there. The biggest employment was glass blowing, and making paper products from the trees growing around us. And I feel very lucky to have escaped this small town of 5,000 people. Actually, nobody else from my town actually even went to college. And so I was the first, and also first in my family to go to college at University of Lund in Sweden. I also caught the science bug early. I actually initially wanted to become an archaeologist, and then when I was 15, I picked up a book on molecular biology. This was back in the early 1980s, actually 1980, I think it was. And molecular biology had just started taking off, cloning and things like that. And I found that really fascinating. And I decided I wanted to be studying molecular biology. And so then I went to University of Lund and studied biology, biochemistry, and biotechnology. And then I got the opportunity to come to the United States for an exchange program at UC San Diego. And I found it so enticing, and I felt like I've been born in the wrong place, really. Because everything was so open, and folks really treated you like an equal in some ways. Whereas in Europe, it's much more hierarchical in academia, and you could do anything in the US, and that was so exciting. So I decided to stay for a PhD. So I never went back to Sweden to continue my education there. While a PhD student, I was in a ginormous lab, 50 people on 2 different floors with Michael G. Rosenfeld. And yet, Jeff Rosenfeld, my PhD advisor, is just this amazing person who had this enormous passion for science. And so I met with him pretty much every day, even though I had 50 people in the lab. And the energy was just incredible. And so I felt I really wanted to do basic science, which we were doing at the time. And I have actually never thought about entrepreneurship at all. And that continued even as I was a postdoc with Robert Tejan. Everything was basic science, just figuring out the fundamentals of what happens inside of a nucleus, how a gene switch on and off. And I felt like that was really exhilarating to discover new things that had not been seen before, like transcription co-activated complexes. Then I went on and accepted a professor position at Harvard Medical School in the cell biology department, and also at the Massachusetts General Hospital Cancer Center. And there I realized that biomedical science with applications, that kind of opened my eyes to that at the hospital and at the medical school. And so I started thinking along the lines of maybe some of the research that we're doing could actually be used to help find new medicines. And so my research started more and more to be tailored towards understanding molecular mechanisms, but with the lens of eventually finding therapeutic avenues. Initially, we studied transcription factors, so genes that are switching, or proteins rather, that are switching genes off and on. And we wanted to drug them, but then we realized that's really, really difficult. And serendipitously, about 15 years ago, still at Harvard, my lab discovered another class of gene regulators called microRNAs as key regulators of gene output. And we realized that microRNAs, the tiny snippets of regulatory RNA, they're about 20 nucleotides long or so, and you can actually find something called antisense oligonucleotides that bind in the base pair, Watson-Crick base pairing manner to the microRNA and gums it up. So it's basically like molecular Velcro. And antisense therapeutics were well described already at the time, but not really for studying or for drugging microRNAs. And we set out then in my lab, basically my lab turned into a biotech incubator. So we set out to drug a couple of microRNAs that we discovered using this antisense technology. And nobody really successfully done this before, and it was really a daunting challenge. So over the next 10 years, first we realized that microRNAs are really not well conserved across species. And that's why there haven't been anything approved as a drug targeting a microRNA, because many microRNAs, even though you may find a pathological role, let's say in a mouse or a rat, that doesn't translate often to humans because microRNA function are often not conserved. They're kind of agents of evolutionary adaptation, actually. But we use human genetics to identify microRNAs that play a key role in human physiology and disease. So with that lens first, and then getting access to human tissues and to plasma and validate that the microRNA is elevated in patient samples, then we went into animal models and actually validated that the microRNA also plays a similar role in the animals. And so the microRNAs that we targeted, they're called MER33 and MER128. We found that they play key pathological roles in cholesterol regulation, in obesity, type 2 diabetes, fatty liver diseases, dry age-related macular degeneration, and Duchenne muscular dystrophy. Quite remarkable. Just 2 microRNAs play key roles in all of these diseases. We published in Science, Cell, Nature Medicine, Science Translational Medicine. These are really amazing therapeutic targets, and they're conserved from mice to humans in their pathological role. So then I decided I'm going to spin out a company focused on targeting these 2 microRNAs. And so about 2 years ago, I spun out this company called Elenae Therapeutics, E-L-E-N-A-E. So E-L-E-N-A-E, you pronounce it Elenae because it's the acronym for locked nucleic acid antisense, so LNA. And so we are using this technology. It's a chemical technology that affords much increased affinity and stability on antisense oligonucleotides. And it's, in my estimation, state of the art of antisense technology. And so we're using those short oligonucleotides to target these pathological microRNAs, initially focusing on Duchenne muscular dystrophy because of the huge unmet need. There's really nothing that works for these poor patients. It's a rare disease. 1 in 5,000 young men are afflicted with this X-linked disease, and exon skipping antisense or AAV microdystrophin doesn't really work very well and are incredibly expensive. And the patients die in their 20s and 30s. So there's really nothing out there for them. And we realized that one of our microRNAs called MER128 plays a key pathological role in the etiology of the disease. We haven't published on this yet. It's we're actually preparing this for Nature, but to be submitted in a few weeks. So it's quite exciting development. We showed that this is conserved in zebrafish, in mice, and in pigs, and of course in humans. So we have data from all of those organisms in one paper showing the pathological role of this microRNA and how we can therapeutically target it. Incredibly enough, the same microRNA is also implicated in heart failure. And so we decided to target it also after myocardial infarction, and we can cure heart failure with dilated cardiomyopathy in mice as well. So if you have a heart attack, we give the mice in the ambulance, so to speak, an injection of our antisense, and they're cured, which is quite amazing. So the company was formed. The problem with starting a company in academia, in my view, is that at least in the last year, it's been incredibly difficult to get money. There's been a complete meltdown in biotech financing. And if you're going out on a limb and doing something that nobody else has done before, first, targeting microRNAs, it's never been successfully done. Antisense, a lot of folks think it's only for rare disease and it's toxic. And I have not been able to convince a single venture capital that this is something that's actually going to fly. We still only have angel investors. And I'm actually realized that this is an impossible task of being a full-time professor and trying to spin this out, this very difficult concept of starting a company around a new technology that nobody's ever done before. So I decided to to take a leave of absence. I'm actually now the full-time CEO of this company. And I moved the company into the Baker Labs incubator, and we're just about to take off. And so it's pretty exciting times. So I'm doing the dog and pony show for venture capital right now. Unfortunately, you have to kiss many frogs before you find a prince. So that's quite a daunting task. But one of the key things that I think is going to be really important going forward is finding advisors. People have done something like this before. I've never done it before really successfully. And so I'm getting advisors now through the Baker Labs, so mentors who can actually help me navigating all of these different things like fundraising, setting up a company from scratch. It's really daunting. I'd like to go back a little bit. I actually wrote down some notes before, and I forgot to mention them as I babbled away here. One of the key things I think when you're starting a company, and it was mentioned earlier, is patents. And I'm not sure that it has been emphasized enough, but having the IP before you start a company is absolutely critical. You can't really develop that effectively in the company. You have to have something to stand on. And it's also first to file now since 2013. So the earlier you file in academia, the better. So so no investors will look at your company unless you have some IP. In my experience, another thing that was mentioned is collaboration. To get to the point where we had all that animal data, as Nabanita said, you have to collaborate, because I didn't have any clue about Duchenne muscular dystrophy. We started collaborating then with Lou Kunkel at Harvard, who actually discovered the dystrophin gene that's mutated in Duchenne muscular dystrophy. And so he helped us out all along the way, and now he's the chair of my SAB. And so that kind of collaboration is really essential. Also, another thing that I wanted to say is you need to gather a team. And every time I talk to folks when I was starting this company is on the venture side, so who's full-time in the company? And I said, well, I don't have anybody right now, but if you give me money, I'll hire a team. And they would just say, no, you have to find somebody who's driving the company. I didn't have a postdoc who could jump at the time, and because she was still finishing all her research and she didn't want to leave for my company. Actually, now she's on board on the company full time. But that's really essential to find somebody who can drive the company. Other things, never give up. Persistence is key. I've been at this for now a decade to incubate this company in my lab. And then now finally I started it, I'm the CEO, and it's really hard all along the way. You have so many hurdles, things go wrong, you just can't give up. You have to be persistent. Another last thing I wanted to say as part of my list here, we mentioned different resources at UC Berkeley. So yes, BBH, LSEC, Baker Labs, Skydeck, all that is fantastic. There are also other incubators like Y Combinator, IndieBio, for example, that can be really helpful for young startups. Lastly, I'd like to mention Nucleate, something that I think is really helpful as well. So it's an academic for students and postdocs, basically, to start a company spinning it out of academia. And actually, I have another company that I'm just about to start that is a Nucleate finalist, and we're actually in the 2023 cohort right now. And it's really fun to work with that group. And I strongly encourage folks to try to apply to Nucleate. Fantastic group. And I think I've been taking too much time here, and I'll open it up for questions. Thank you.
This is awesome. Thank you, Anders, for laying it out raw. I think it's important for everybody to understand that it is not easy for sure, but what is easy in life? And postdoc was not easy. Coming to this country was not easy, which I think most of the people in the audience are, I think. I'd like to go something more very, very fundamental, because you're a basic researcher and exploring the science of a cell, what's inside it. And then a lot of the postdocs are on campus and across the country complaining about the same thing, that, hey, I'm doing basic research. What did it take for you to think about entrepreneurship? I know you're at Harvard, and is it talking to patients, or like what exactly happened there? If there's one or two takeaways of how people should think about basic research and that translation part of it.
Yeah, I think it was the environment at Harvard Medical School and Massachusetts General Hospital. I was surrounded by people who thought about biomedical and patients and applications all the time. And it kind of changed my thinking from just doing basic science into applications. So yeah, if your professor doesn't have that bent, you're going to be alone. So you need to really find like-minded people around you. And I think this organization that's hosting me right now is one such organization. I think Nucleate and iCorps and many other entities on campus can help you, even if your professor is not necessarily entrepreneurial. It certainly helps, of course, to have a professor that's entrepreneurial for you to spin out a company, but it's not necessary.
Yeah, you're a brave heart. I mean, to take a leave of absence, become a complete full-time CEO. I know some of the participants here are trying to be faculty. What would that message be for them to think about more entrepreneurially than just to publish and do the research? Not that it is not important to do basic research. I think the translation part is not emphasized really hard, unless you get a good advisor like Bob Tijan. Great, like he started companies. I think that's a great inspiration. But I think the next generation of faculty should think about that way.
Yeah, no, a lot of times, when you're younger, you're junior faculty, you can't just pick up and start a company, because you're focused on getting tenure, getting publications, grants, and all that stuff. And it's very, very difficult. I think for somebody more senior like myself, it's much more natural, I think, because you're starting to think, so what else? I have many publications and I have done a lot of things, but I haven't done a startup. So that's kind of the thinking for me. It's like I wanted to do something else, especially something that can help patients. You have to have a great idea. You have to have something that you believe in, maybe a 10x solution, not just build a better mousetrap, but something completely out of the box. That's at least my view. Otherwise, for me, it wouldn't be worth it. And I felt microRNAs, it's a completely untapped area, and nobody knows really about it. I talked to venture capital, and they're like, what's that? And that excites me, that it's so new and so different. And the antisense technology that we're pursuing hasn't really been applied effectively to these types of diseases, just a few rare diseases. And if we can impact, let's say, obesity, type 2 diabetes, cardiovascular disease, and so on, using this technology, that could be tremendously impactful.
Thank you. That was really insightful. And I loved your summary in the end too. I think that really hit home in a lot of ways. I did see Yi had his hand raised. Yi, do you have a question? You want to go first? We also have a list of questions, and so I'd want to call upon everyone who's here to ask their questions first.
Sure. The question I wanted to ask maybe has been partially answered by the previous questions. So the question is for Professor Naar. Thank you very much for sharing your stories. So my question is that, like, at the very beginning of this conversation, the other two panelists mentioned about the risk, right? On the other hand, as a tenure professor, how that tenure cushion will change your taste about the risk. And on the other hand, you also mentioned that you will make a decision whether you want to pursue a different thing in the entrepreneur or keep your kind of professorship still going on with the same everyday life. So you mentioned some thought like you want a difference and a different life. And I also want to ask a more maybe a personal answer. Let's say if one day you were like thinking about when it was a day you were thinking about, should I keep the same professor life or entrepreneurship lifestyle? Like, how does that affect your, let's say, your family or your like everyday work style? So like not about just like very high level answer, like I want a different lifestyle. I already tenured. What I still can get, but more personal or detail oriented answer. Mm-hmm.
Yeah, so it's a completely different way of thinking about work. So before it was, you do grants, you write papers, you teach, and it goes round and round and round like this. Nothing really changes. But starting a biotech company, everything is new all the time. And I find that exhilarating, and it's much more exciting than the grants, the papers, and the teaching, honestly, right now at least. Maybe ask me in a year when the company completely tanks and everything is, nothing is working. But right now it's a very exhilarating time, and I've put in many hours working on this, to my family's detriment, I guess. You have to sacrifice something when you're doing these kind of entrepreneurships.
Yeah, I think you learn a lot though in this process than in academia. It's just like so many hats that you're putting. Just kissing those frogs. Maybe that's a good segue to talk about kissing those frogs. Maybe it's a common question to Nabs and Adam as well. Talk about the lessons you learned while talking to this venture capitalist. What is working? What is not working? What are the lessons you've been walking away with?
Right. Yeah. So as I mentioned, I think the biggest challenge right now is the financing falling off a cliff. And so everybody is super conservative. And if you come to them, to venture capital, with something totally off the wall that they've never heard about before, the challenge of convincing them to put millions of dollars on the line for something that's completely untested, it's a very daunting challenge. And if you're doing, I would say AAV, as we heard from Adam, everybody knows what AAV is. Yeah. And if you do, let's say, AI or CRISPR, it's much easier, I think, to find folks that can jump onto the bandwagon, kind of in the Me Too approaches. Mm-hmm. But for something that's completely out there, it's really, really hard. That's my experience.
Yeah, I think Dave Schiefer was in the similar boat 15 years ago when he was talking about gene therapies and AAVs, and nobody was willing to fund. He mentioned that several times, but persistence, I think. Yeah, as you mentioned earlier, that is important.
Yeah, Dave was in that similar boat, and you really need people like Dave and doing those things that are completely off the wall, brand new, because that's how entire new fields open up and areas of investment and impact on patients' lives. I think, I have it a little bit easier because we have a platform, that, like the viral platform, it's been well established, and we have the one off the wall kind of brand new thing that differentiates us, but we're combining it with like clinically approved modalities. That makes it a little easier than if the whole thing is completely brand new. But it's not to say that those completely new things aren't extremely important, because that's what opens up these new areas to do, and somebody's got to do it. So
Naps, what has your experience been with investors?
I think the current fundraising landscape also makes a very big difference in our journey. I think biotech investment is also at an all-time low because of how the market's doing, and most of the investors are looking to add more money to their portfolio companies who don't have a lot of runway anymore. So they are extremely conservative. They also require a lot of de-risking of the science, and obviously, and that requires a lot of capital too on our end. So I think it's been a hybrid approach of looking at a lot of grants and non-dilutive money in order to build a data package that can convince a sophisticated biotech investor.
Radhika, you have any questions that you previously received?
I think one of the main ones that sort of pops up is, how do you assemble your first team? And how do you go about choosing the right people? So, if you have thoughts about how you're planning or how you're already doing it, that'll be.
Yeah, well, for me, I think that's one of the big advantages that you have when you're spinning out of the university from a lab, because like I said, one of my co-founders was a postdoc who we had previously collaborated on an academic project. And that really de-risked it, because we knew that we had complementary skill sets and we knew that we worked well together, and that he would be a good fit for the culture and everything. Likewise with one of the other people involved at the bench, this was someone who was working for me, who I was mentoring. And so again, we had that chance to de-risk. I think, I think that's been a really helpful part of spinning out of academia. And I think that the early team that you bring on, at least so I'm told, is pretty crucial for setting the tone and the culture of the company going forward. But we're in the early days, so it's TBD.
I think one for us, well, similar to Adam, the founding team was from the people who worked on the technology. So we've known each other worked together for many years, seen ups and downs, mood swings, and gotten to really know each other through thick and thin. So it wasn't, that was, I guess, how we de-risked the founding team. But moving forward, the other people who got involved, it's not just on employee level, but just more involved in the company and in the cap table, such as like, a new board member, executive chairman, and so on. We made sure that we built a relationship for a good period of time. For example, with our executive chairman, he met us for, way before, just to talk about the science, even when we were just thinking about spinning out the company, but didn't really have any plans, haven't incorporated, really didn't think about the business. It was just about the science. And he kept up with it for a very long time before we decided to really formalize that relationship. And I thought that really helped, because we got a feel of who we're going to be working with, and probably for a long time. And similar to that with the other few members who joined as well. At least a couple of months of getting to know each other for a while, seeing what kind of value they bring, and also the kind of whether you like working with that person or not. I think that's also really important.
Yeah, I'd like to echo all of those comments as well. And for me, given the difficulties in getting funding, and also convincing venture capital and folks around me to support this new, completely new crazy idea, I would say, what I found was helpful is to recruit consultants that can actually be a very economical way of getting not employees, but folks that have industry experience and that can help you along the way. And so I got one consultant that I paid with some of my seed funding to create a business plan for me. Another consultant is basically working as a chief scientific officer. She has another day job, but she's moonlighting for my company. And she has decades of experience with muscular dystrophy therapeutics. And so that's a very economical way of getting experience without actually committing to having to pay their salary and to share your equity at an early stage before you even have the company up and running. So especially at this day and age now, when financing is so stretched, I think consultants is a good way to go.
How are the investors reacting to that, having consultants as opposed to full-time? Because venture capitalists are venture capitalists. I don't think they have a heart. they have a brain that thinks about money, and it's an egg or chicken problem here, right? Like without money you can't hire. What are you thinking about that process? When you go to an investor, they're looking for that team.
Right.
How are they reactive to consultants?
It's still a work in progress, because a lot of these consultants just came on over the last few months. So I haven't really gotten a lot of feedback from the VCs, what they think about that. What the consultants have brought to the table is maturing of our vision. So they can actually bring the industry experience and to flesh out the plan going forward. So for example, for Duchenne muscular dystrophy, as I mentioned, we have all this data. What are the next steps? Next steps is CMC. So basically you need to make a drug, and I have no idea how to make a drug. And so I hired folks that have done it before as consultants. So they fleshed out a plan for me. This is how you make a drug for Duchenne muscular dystrophy. And so now I can present that to venture capital. There's no way I could have done that myself. Maybe even hiring people, the right people, would have been very difficult.
Yeah, I want to open the floor to anyone else who wants to jump in with a question. You can even put it in the chat, and I'm happy to read it out. Or
So I have one more question. So Professor Naar, you mentioned that the financial situation is not very ideal in this period of time, especially for the crazier idea that there is no scientific convention. But according to what I learned in your talk, so that you just want to pursue that one, right? So how can you just find a detour? For example, you mentioned about patent or paper. For example, like you said, you have published Nature and Science. In terms of utility of convincing the investor, like, can you describe a little bit, like, what are those things that those inventor may be looking for in your case, where the idea tends to be of a less convention?
Yeah, that's a good question. Publications really help, for sure. And for the Duchenne muscular dystrophy project, we haven't published on it yet. So we're about to submit a paper to Nature, and hopefully we'll get in, who knows. We're probably going to put it on bioarchives, because I would like to get it out for venture capital and others, of course, the patient community, to see that this is in the pipeline, and to build some excitement around it. Also, I'm an invited speaker at the Muscular Dystrophy Association annual meeting coming up next month. And so I'm excited to share this, I think, very significant advance with the community. And that would then build, I think, excitement around this.
Have you been applying to SBIRs? And I'm asking this because that's the other wing of fundraising for the early stage and risky ones, because this is not widely accepted. How have they been reacting to funding, if you have applied for any?
Yeah, so we were in the process of applying for an SBIR. We submitted one on an unrelated indication that I can't talk about, but we're planning for Duchenne as well. Philanthropy can also play a role. I started another company that I haven't talked about. When the pandemic started, we decided to pivot our antisense technology to target SARS-CoV-2, and we have developed a nasal spray that completely prevents the virus from replicating. We published that in Nature Communications last year, and I started another company around that called Calgen. It's been very difficult for a different reason to get funding for that from venture capital, because all of them think that, well, the pandemic is surely over.
It's gone.
It's around the corner, it's going to be over. And their time horizon is longer, and so they don't believe that we'll have a drug, a nasal spray, in time to have an impact on the pandemic. So the way we got money for that is actually through philanthropy. So I got $2 million from one billionaire to help develop this further. And I'm flying up to the Gates Foundation on Sunday to talk to them about another $1 million. And so this is philanthropy, which could help in if the indications is right. If you can entice some philanthropic organization to pitch in, that could be another avenue.
Great point. Great point. I don't know, for some of you, if you're looking for some stories, I have a podcast called Lab to Startup, where we have a bunch of founders who went through such long process and survived and are successful now. Check it out.
Right. Sarvesh, do you want to ask your question? You want me to read it out?
Okay.
All right. So he wants to know, how do we convince investors when you have no IP, but you're doing something that's quite well known, but with the potential in a different geographical location?
That sounds really challenging.
That's probably tailored to a different set of investors. Like, so a biotech focused investor definitely needs IP and probably won't be looking into other geographical locations as much. If we got a bit more context about what you're talking about, Sarvesh, I think we'd be able to answer this better. But yeah, but
Yeah.
there's a lot of different types of investors. So you just have to figure out who would be interested in a technology like that in a different geographical location.
So thank you very much for the answer. So the question is about if you want to do enter into a manufacturing market, basically. And there are very many different, very well-known techniques there. And then you want to just pick one, and because there is a huge market and there are not many companies available there, and then you just want to invest, you want to build up your company there. In such a scenario, how do you attract people to invest in that?
That it depends on the type of company and the market. If you take a bike company like Lime, or there are so many investors that put money into different markets because there's a different market size, and
Okay.
so it doesn't really have to be IP. If you have the right execution team and a plan for that and a big enough market, yeah, deep tech, I think, like biotech and those kinds of things, IP is really important. But if it is a different sector, things work differently, right?
Manufacturing IP can be a little bit more difficult to like protect, because like some people in manufacturing space, and I'm speaking from a position like in the gene therapy manufacturing space, will actually choose to go trade secret because they don't want to put out like the secret sauce per se for the world to see, just because it's so difficult to litigate and protect, like stop other companies from using that. It's a lot easier to protect IP when it's like in the end product, like the FDA approved product that goes through like the, that's the actual like product going into the patient. So I guess if you had like some sort of way where you can manufacture much more cost effectively, like in the case of AAVs, they're extremely expensive to manufacture and they can't manufacture them enough. So even something like a 2 to 5-fold increase in production would be significant. Then it could work. But I guess those are some things I'm thinking. Some people keep trade secrets when it comes to manufacturing.
So just taking one example, say there is one turbo pump, say in US, that, or like most of the companies are in Germany, that you get it for like 25K, and then you take it to a different location, say Africa, and there you have the cheap labor costs, but you have a very good team. They are very good skilled at what you are planning to do, and then you can set up a team and then reduce the price of that particular pump to say 10K. So if instead of from 25K to 10K, if you are able to do that, but you have to how to show that, like, yes, we will be able to do it, and then get the investors to invest in that.
My guess is you'd probably have to have somebody who had actually been involved in doing that before, And then you'd also have to make sure that this pump that you're manufacturing is like, it itself is off patent, or that, it's a generic thing that you're freely able to operate upon.
All right. Thank you, everyone. Just want to be cognizant of everyone's time. I like to ask this. What's your best thing about what you're doing, which makes you wake up every day?
Yeah, the excitement, the potential to make a difference, to help patients, it's a pretty amazing feeling when that potential is there. So
I think I echo Adam's comment. And also being able to drive that change, to see that you could be leading something that could help so many people. Could probably take a lot of time and a lot of work, but at least we're making some progress there. I think that's what helps us be entrepreneurs and make a lot of sacrifices along the way.
Awesome. Thank you so much for sharing all of that. And on that happy note, I'd like to thank all our 3 speakers. This has been a fun session, and everyone for joining us.
Thanks for having us.